Cancer, the leading cause of death in Canada. Since 2007, Statistics Canada has reported that cancer is responsible for approximately 30% of all Canadian deaths. Cancer cells are characterized by their ability to grow out of control and become invasive. Unlike healthy cells, cancer cells do not mature into distinct cell types with specific functions. They are able to ignore the signals that prevent abnormal cell growth and apoptosis. For these reasons, they are able to divide continuously and form a tumor. These malignant tumors are able to spread or metastasize to other parts of the body. This can severely impact the way the body functions and often results in death. Cancer is considered to be a genetic disease. While it can be inherited, it can also be caused by errors in cell division or DNA damage from environmental exposure. These mutations often occur in proto-oncogenes, tumor suppressors, and DNA repair genes. Despite the years of research and all the technological advances in science and medicine, we have still not been able to cure cancer in humans. Given the complexity of a disease like cancer, a deeper understanding of the way healthy cells transform into cancerous cells may help us in developing novel therapeutic strategies. As cancer cells develop from normal cells, many cells experience changes in metabolism. Metabolism can be split into two core processes. Catabolism, or degradation, is the breakdown of nutrients such as carbohydrates, proteins, and fats. This process provides reducing equivalents, oxidized metabolites, and energy in the form of ATP. These products are then used in the other core metabolic process, anabolism. Anabolism results in reduced metabolites and is the biosynthesis of essential cellular components such as DNA, the phospholipid bilayer, and structural proteins. The enzymes catalyzing the reactions in these opposing processes are tightly controlled by regulators, compartmentalization, and feedback inhibition to meet the body's changing energy demands. The major catabolic processes that occur within animal cells are simplified and shown here. The breakdown of carbohydrates, fats, and proteins begin in separate pathways but converge at the mitochondria. In the mitochondria, intermediates of these pathways enter the tricarboxylic or TCA cycle. Reducing equivalents produced in both the cytosol and TCA cycle then deliver their electrons to the terminal electron acceptor, oxygen, in the electron transport chain. This process of oxidative phosphorylation results in the generation of high amounts of ATP. A change in the metabolism of cancer cells was first reported by the German scientist and Nobel laureate Otto Warburg almost 100 years ago. He found that cancer cells take up a lot more glucose than healthy cells and produce significantly more lactate. Glucose is a carbohydrate, and the pathway for carbohydrate metabolism involves glycolysis. After glucose enters a cell, it undergoes a series of reactions catalyzed by various enzymes in the cytosol. Each reaction produces the intermediates shown here, which are subsequently used in the next reaction. Ultimately, the process of glycolysis converts one molecule of glucose to 2 molecules of pyruvate and generates 2 ATP and 2 NADH. In healthy cells, the pyruvate from glycolysis has 2 fates. When oxygen is present, oxidative phosphorylation can occur. So the pyruvate enters the mitochondria and is completely oxidized to produce carbon dioxide and acetyl-CoA. Acetyl-CoA enters the TCA cycle, and the reducing equivalents from glycolysis and the TCA cycle deliver their electrons to the oxygen in the electron transport chain. This generates 36 moles of ATP. When oxygen is not present, oxidative phosphorylation cannot occur. The pyruvate molecules do not enter the mitochondria and are reduced to lactate instead. This produces 2 moles of ATP. In proliferating cancer cells, Warburg noticed that in the absence or presence of oxygen, pyruvate was mostly converted to lactate. This generates 4 moles of ATP. This preferential lactate generation alongside high glucose uptake and increased glycolysis flux is known as the Warburg effect, or aerobic glycolysis. Warburg initially believed that the diversion of pyruvate to lactate was due to a defect in the mitochondria of cancerous cells. He even hypothesized that this defect was the cause of cancer. However, later studies have shown that the mitochondria of cancer cells are not defected, therefore aerobic glycolysis cannot be attributed to this. In fact, it is now believed that the Warburg effect is not merely a consequence of cancer, but a regulated mechanism that occurs to provide some benefit to cancer cells. This brings us to our research question. What role does the Warburg effect have in proliferating cancer cells? Why would these cells choose to inefficiently generate ATP through glycolysis, even when oxygen is present? Although Warburg made his discovery almost a century ago, interest in this topic has only picked up in the last 10 years. Since then, a number of proposals have been put forth to explain this effect in cancer cells. One explanation states that the Warburg effect occurs to promote biosynthesis in proliferating cancer cells.
Cancer cells require various types of precursor molecules and high amounts of the reducing equivalent NADPH to synthesize macromolecules necessary for proliferation. These include nucleotides for genome replication, amino acids for proteins, and lipids for membranes. The glycolytic pathway provides a major portion of the required precursor molecules in the form of intermediates and branches into other biosynthetic pathways that generate NADPH. So how exactly does the Warburg effect promote biosynthesis pathways? The Warburg effect decouples oxidative phosphorylation from glycolysis and ATP generation, allowing for high glycolytic flux. But this glycolytic flux can be inhibited by NADH accumulation in the cytosol, as the electrons are not being donated to oxygen. The reduction of pyruvate to lactate regenerates NAD+ to be used again in glycolysis. This prevents the inhibition of glycolysis by NADH. In this way, the Warburg effect is believed to contribute to biosynthesis by increasing glycolytic flux, which in turn increases levels of glycolytic intermediates that can be used in the branching biosynthetic pathways. One of the first intermediates of glycolysis, glucose 6-phosphate or G6P, is involved in the pentose phosphate pathway or PPP. G6P is used to generate NADPH required for reductive biosynthesis and ribose 5-phosphate, a major component of nucleic acids. The PPP is comprised of an oxidative and a non-oxidative phase. The oxidative phase produces NADPH molecules, while the non-oxidative phase results in the generation of ribose sugars and other glycolytic intermediates. The first rate-limiting enzyme of the PPP is G6P dehydrogenase. This enzyme is responsible for oxidizing G6P to 6-phosphoglucanolactone, thereby resulting in the release of NADPH. A study investigating the effects of p53 on the PPP has shown some interesting results linking human tumors and the PPP with regards to G6PD. The p53 tumor suppressor protein activates DNA repair enzymes, arrests cell growth, and initiates apoptosis. Mutations in the p53 gene is associated with most cancers. In the first figure, the oxidative PPP flux in human colon cancer cells was assessed by measuring glucose consumption. p53 deficient cells had significantly higher levels of oxidative PPP flux compared to controls expressing p53. To elucidate the mechanism behind how p53 deficiency upregulated PPP flux, they investigated the effects of p53 on the activity of G6PD as shown by Figure 2. G6PD activity was assessed by measuring rate of NADPH production in human bone osteosarcoma epithelial cells. A twofold increase in G6PD activity was observed in p53 knockout cells. Silencing p53 did not alter G6PD total protein expression levels. This suggests that the G6PD increased activity due to the lack of functional p53 contributed to increased PPP oxidative flux. Together, these results show most cancers devoid of functional p53 have higher G6PD activity and higher oxidative PPP flux. This suggests that the Warburg effect in proliferating cells may be necessary to provide the precursor molecules required for biosynthetic pathways such as the PPP. In the non-oxidative phase of PPP, glycolytic intermediates such as fructose 6-phosphate are regenerated and fed back into glycolysis. This phase involves the key enzyme transaldolase, or TALDO. TALDO converts cetoheptylo 7-phosphate and glyceraldehyde 3-phosphate to fructose 6-phosphate and erythrose 4-phosphate. F6P is able to re-enter glycolysis, while erythrose 4-phosphate is used to synthesize aromatic amino acids. A study from the Fudan University in Shanghai investigated transaldolase as a biomarker of hepatocellular carcinoma, which is the most common type of primary liver cancer and third most common cause of cancer mortality. The first figure shows TALDO expression levels in metastatic and non-metastatic human colon cancer cell lines. The metastatic cell lines 97H, 97L, LM6, and LM3 showed significantly higher TALDO expression levels than the non-metastatic cell lines. This suggests that TALDO expression may be necessary for metastasis. Figure 2 shows serum TALDO levels in metastatic and non-metastatic HCC patients. Results from these figures suggest that TALDO is upregulated and required for the growth and spread of cancer cells. Taken together, upregulation of key enzymes of the PPP to produce necessary cellular components would require higher levels of precursor molecules. By increasing glycolytic flux, the Warburg effect allows for high levels of G6P that can be used in this biosynthetic pathway. The next glycolytic intermediate that branches out into a biosynthetic pathway is dihydroxyacetone phosphate, or DHAP. Glycerol 3-phosphate dehydrogenase uses DHAP to produce glycerol 3-phosphate, which is then used by fatty acid synthase, FAS, and other downstream fat synthesizing enzymes to produce lipids. Since lipids are a major component of the phospholipid bilayer that make up cell membranes, glycerol 3-phosphate is essential to proliferating cells. In this study, researchers showed that the lipid biosynthetic pathway involving the glycolytic intermediate DHAP was upregulated in human bladder cancer. They used the rate of NADPH consumption to measure the activity of various lipogenic enzymes. This figure shows that glycerol 3-phosphate dehydrogenase activity was significantly increased in tumor cells when compared to normal healthy cells. The next two figures show the activity of other enzymes involved in fat synthesis. FAS, which directly utilizes glycerol 3-phosphate, is shown to have increased activity in tumor cells. Figure 3 shows the activity of downstream enzymes that utilize the products from this reaction to synthesize fatty acids that are also upregulated in these cancer cells. The upregulation of the rate-limiting enzyme glycerol 3-phosphate dehydrogenase would require higher amounts of the glycolytic intermediate DHAP. Increased glycolysis due to the Warburg effect would allow for this DHAP accumulation. The increase in activity of downstream lipogenic enzymes alongside glycerol 3-phosphate dehydrogenase suggests that the glycerol 3-phosphate produced in glycolysis is in fact used by proliferating cells to synthesize lipids. Another pathway that branches from glycolysis is the biosynthesis of serine from 3-phosphoglycerate. Serine is required for the synthesis of amino acids cysteine and glycine, where glycine is then incorporated into purines. Serine is also used in folate metabolism. The first rate-limiting step of the serine biosynthesis pathway is the oxidation of 3-PG to 3-phosphohydroxypyruvate by the enzyme phosphoglycerate dehydrogenase, or PHGDH. In 2011, a study from MIT looked at the effects of PHGDH on glycolytic flux and oncogenesis. Figure 1 shows the effect of knocking out various genes encoding enzymes in the serine biosynthesis pathway in esophageal squamous cell carcinoma cell lines. The genes were silenced using short hairpin RNAs. Stable knockdown of PHGDH significantly reduced cell proliferation compared to the control cells. Knockdown of the other enzymes of the serine pathway also reduced cell proliferation. The key finding from the study suggests that PHGDH is required for the proliferation of cancer cells, which extends to support the requirement of the serine biosynthesis pathway for proliferation. Therefore, the Warburg effect is required to continuously supply 3-PG required for biosynthesis of serine in proliferating cancer cells.
The data from the 4 independent studies we have discussed all provide evidence for the upregulation of biosynthetic pathways in proliferating cancer cells. The pentose phosphate pathway, glycerol 3-phosphate, and serine biosynthesis pathways all branch from the central glycolytic pathway in carbohydrate metabolism. These pathways ultimately lead to the formation of vital cellular components such as DNA, cell membranes, and enzymes. The evidence we provided suggests that the Warburg effect allows for the upregulation of these pathways to be sustained by providing high amounts of glycolytic intermediates for use in biosynthesis. Altered metabolic changes have been reported to be a hallmark of various cancers. These reprogrammed metabolic states are emerging as highly regulated processes maintained by cancer cells to promote their survival. For these reasons, targeting the altered metabolism of cancer cells is an attractive therapy solution. For example, inhibitors targeting the overexpressed PHG-DH in serine synthesis are being considered for use in cancer therapy. In fact, some of these inhibitors have been approved, while others are in clinical trials. Our project aims to broaden the understanding of these processes. This may allow scientists to identify inhibitors that target cancer-specific mechanisms and avoid causing toxicity to healthy cells.